In this video tutorial, we're going to teach you guys how to extract plasmid DNA from bacteria. Now, this technique is used often in synthetic biology because we actually rely on bacteria in order to manufacture plasmids for us. After we transform a bacterium with our plasmids, those bacteria will actually copy those plasmids and any genes that are contained within that plasmid and pass those genes on to their offspring. This allows us to clone, which is another word for amplify or copy that plasmid. Once that plasmid is copied, we can then extract that plasmid from bacterial cells, and then we can either test that plasmid to ensure it's assembled correctly, or we can transfer it to a new organism. So there's a lot of different things that we can do with it. So there's 3 steps in plasmid isolation. The first step is actually growing up a culture of bacteria that have been transformed with our plasmid. Once we've grown up a sample of bacteria transformed with our plasmid, we can then collect those cells and break them open in order to release the plasmid DNA. The third step in the plasmid DNA extraction is actually separating the plasmid DNA from the rest of the bacterial cell's components. Usually in molecular biology and synthetic biology, we use kits in order to make that happen. And so we're going to use a kit today in order to extract plasmid DNA. These types of DNA kits are sometimes known as mini prep kits, and they're used frequently. And we buy them from companies. So before we begin, we'll need a few different materials from our GoSTEM kit. From our general laboratory supplies bag, we're going to need a few different things. We're going to need our microtube rack. We're going to need our Sharpie. We're going to need our microtubes, some extra microtubes from that container. And then the last thing, do we need anything else? Nope, that should be enough from that. So that's from our general supplies bag. We'll also need our drill centrifuge that you guys prepared and tested yesterday. So we'll need that. We'll need the rest of our activity number 9 kit. So that's the DNA purification by mini prep kit. We'll need the cultures that we prepared yesterday. So those will be either on your heat block or on your microtube rack that you put on the windowsill, or the tubes that you took out of your pocket that you were keeping warm. Other things that you might find useful are a cell phone or a kitchen timer or something to keep time. And then finally, a pair of scissors. All right, so now that we have all of our materials collected, let's get started. All right, so we're all set up here. So the first step in the procedure is going to be to pellet the bacterial cells from the cultures that we have grown up. So my particular cultures I incubated overnight in my heat block. You might have left yours on the windowsill in the sun, or you might have left yours in your pocket. Either way, you should have nice cloudy cultures, and you should be able to see the bacterial cells. So what we're going to do for this procedure is we're going to put our tubes of bacteria into our drill centrifuges, and we are going to centrifuge them for 2 minutes. So you might want to set a timer. I'm going to use the timer on my computer, and so you're just going to start the drill and let it go for 2 minutes. sound of my timer going off, and so I'll stop my drill. Now, when I take my tubes out, you might be able to see when you look at the bottom of your tubes a small cell pellet. Now, if you don't see it with your naked eye, don't worry, it's probably still there. Okay? So, now what we'll do is we'll discard our supernatant. And so to do this, we can just pour that supernatant into a waste container, like the little beaker that we put in your general supplies bag. Now don't worry, your pellet will remain stuck to the bottom of the tube, so you can just gently shake out the excess liquid. Okay? So we're going to do that for both of our tubes. Now that we've done that, the next step is we are going to have to resuspend that cell pellet in a buffer. Now all of the buffers you need in order to complete this activity are in the little baggie from your activity number 9 bag. Each one is labeled with a different colored tape and has a different number on it. So the first buffer we're looking for is buffer PD1. PD1 is the buffer with the green tape on it. So what we will do is we will take a dropper pipette and we're going to suck up all of the buffer from the PD1 tube, and we're going to put that into one of the tubes with your cell pellets. And then what I'm going to do is I'm just going to suck that liquid up and down and try to put that pellet back into solution. So just kind of do this and just break up that cell pellet. Okay. Once I've got one broken up, I'm going to take that same liquid, put it into the second tube, and again, I'm going to pipe it up and down and try to get that pellet broken up and get those cells into the solution. Okay, very good. So, we have a pipette, we have one tube, and we have the tube of PD1. Okay, those are all empty and those are garbage. So since I don't have a garbage with me right now, I'm just going to use the Activity 9 bag as my garbage can. Okay? So now the next step in our procedure, we're going to actually lyse open the cells. The term lyse means just to burst. And so to do this, we're going to add buffer PD2. Buffer PD2 is a solution with a strong detergent in it. And that strong detergent is going to break open those cells. And so to do this step, we're going to take a new pipette. I'm going to open up the tube of PD2. I'm going to take all that liquid out of the tube. And I'm going to add that to the tube with my resuspended bacterial cells. Okay, I'm going to close up that tube. This pipette and the PD2 tube, those are garbage, so I'm going to put those in my activity number 9 bag. Now for this tube, I'm going to invert this tube gently 10 times to mix. So I'm just going to do this, a nice gentle rocking motion. 6, 7, 8, 9, 10. Okay. So now I'm just going to set my timer for 2 minutes, and I'm going to let that tube incubate for 2 minutes. Okay. Now during this time, the strong detergent that we added to the tube is going to act on the plasma membrane of the cells. Now if you'll remember from maybe your life sciences classes that you may have taken, the plasma membrane is a layer which separates the inside of the cell from the outside of the cell. That layer is made of fats, which we in science we call lipids, as well as proteins. And the strong detergent, it breaks up those lipids and the proteins that separate the inside of the cell from the external environment. And so as we're letting these reactions sit, the cell membranes are being damaged, and the contents inside of the cell are being released out into the rest of the solution. And so this is the lysis step of our mini prep. Okay. For our next step, we're going to have to neutralize the reaction. And so to do that, we're going to add another buffer. The buffer we're going to add this time is buffer PD3. Buffer PD3 is in the tube with the yellow labeling. We don't want to add that buffer until our 2 minutes are over. Okay. We'll also need a new pipette for that. A few more seconds. Done. So now I'm going to add buffer PD3 to my cell lysate. So again, I take a new pipette, I'm going to suck all that liquid up from the tube that says buffer PD3 on it, and I'm going to add that liquid to my other tube. Okay. Now when you add that, you might notice that a precipitate forms. Hopefully you can see that in this reaction. That precipitate is cell debris. So that's good. That's exactly what we want. And if you don't see this, don't worry. Okay? Just because you can't see it doesn't mean it's not there. Okay? I'm just going to set these two things away in my waste bag because we don't need them anymore. And now what I'm going to do is I'm just going to take this tube and I'm just going to gently rock it back and forth 10 times in order to mix the tube really well. 5, 6, 7, 8, 9, 10. I don't know if you can see, but my tube, I can see these big white chunks in it. Okay, that's good. That's the cell debris. Okay. So, now that we've neutralized our reaction, we can proceed with the next step. For this next step, we're going to need our drill centrifuge again. We're going to put our tube with our cell lysate in one side. And then we're going to take just an extra microfuge tube, or microtube, from our general supplies bag. And we're going to use that to balance the centrifuge. And that's really important because we need the extra weight in order to make sure that our drill centrifuge doesn't wobble. So make sure that you have an empty tube to balance the centrifuge. Now this time we're going to centrifuge again, and we're going to centrifuge this time for 3 minutes. So I'm just going to reset my clock here. I can figure out how to do that. Okay. There we go. I can figure out how to do that. Technology's hard when you get old. Okay, and let's start. Okay, so 3 minutes are done. So, when I look at my tube, I see that some of that cell debris has pelleted on the side of the tube. Now, there's still some in solution. I guess my drill doesn't go quite fast enough to pellet that all, but that's okay. We'll make do with what we've got. Okay, so take that. Now, the next step in our procedure is we're going to take the cell lysate and we are going to load it to a centrifugation column, sometimes known as a spin column. Now, the spin column has two components. Okay, there's an upper component, which is our actual column. The column itself opens just like a centrifuge tube does. At the bottom of that column, there's a white resin. That resin binds to plasmid DNA. Okay? And so we're going to bind our plasmid DNA to that resin. We're then going to wash that plasmid DNA clean, and then we're going to cause the plasmid DNA to separate from that column, and that's called elution. The other part of the spin column is a collection tube. The job of this tube is just to collect any liquid that flows through the column. Okay, so make sure you use the column and the collection tube together. Okay, so the first step in this procedure is we're going to take a pipette and we are going to try to collect the supernatant from our centrifugation and separate it from the cell debris. Okay, now I don't know if you can see that in my tube, but I had a little bit of trouble because my drill, the battery must be dying, and so it didn't go fast enough in order to separate the cell debris, which is the white precipitate, from the liquid. Okay? So what I'm going to do is I am going to go and get a Kleenex. So I'm going to pause recording on the video while I go do that. And then I'll teach you guys how to separate that cell debris. Okay, I'm back. So I've got my Kleenexes, or what are the equivalent of Kleenexes in the laboratory, Kim wipes. If you don't have any Kleenexes or, facial tissue, you can use a piece of toilet paper too. So just any sort of disposable paper. What we're going to do is we're going to use this as a filter in order to filter out those bits of cell debris. And so what I want you to do is to take your Kleenex or your facial tissue or your piece of toilet paper, and I want you to fold it into a triangle and then fold that triangle again. And that will create a little pirate hat structure. If you open up one of those folds, now we have a little cone. So I'm going to take that little cone, and I think for right now, I'm going to put that into a new micro tube, okay? And I'll hold it just like that. So this is what I'm doing, guys. I'm going to take that supernatant with the debris, and I'm going to put that into that little cone, and then I'm going to squeeze that paper in order to separate out the liquid from the rest of the cell debris. You'll lose some of the liquid in the process, but that'll be okay. Now, I wore gloves from this just because I have gloves that are easily available, and I didn't want to get this stuff on my hands and then have to go wash my hands because I'm filming a video. If you have gloves, you can do the same thing. You can wear a pair of gloves. You're going to squeeze that paper really hard in order to get the last drops out. But if you don't have gloves at home, that's okay. You can do this with your hands. And then just make sure that you pause the video and you go wash your hands with soap and water for 30 seconds right away. so that you don't get any of the stuff from the kit or from the bacterial cells into your mouth or your eyes. Okay? So again, I'm done with the gloves, so I'm just going to throw them away. And if you don't have gloves, don't worry about it. So now, as you can see, the stuff in my tube, I filtered it. And so none of that nasty white cell debris is still present. Okay, I lost some of my liquid, but that's just part of the deal. Okay, the rest of my pellet from my tube, I can throw in my waste bag. So now I'm going to take the filtrate, which is the part of anything that you filter that runs through the filter, and I'm going to load that onto my spin column. Okay? So I'm going to take my pipette here. I'm going to suck up all that filtered liquid that should contain my plasmids, and I'm going to put that into my column. And so I'll just squeeze it on into there. Now I can close my column. Okay. This pipette, I'm going to set that aside. This one tube, now that it's empty, that can go in my garbage. Now I'm going to take my spin column and I'm going to spin it in my centrifuge. So I've got my balance tube right here. I'm going to put my spin column right there. I'm going to come over to my timer. And I'm going to set my timer for 2 minutes. And I'm going to centrifuge for 2 minutes. Timer. Alright, so what you might notice is that some of the liquid has gone through my column and it's collected down in the bottom of the tube. So that's good. That's exactly what we want because that liquid contained our plasmid when it was up in the collection tube. And as that liquid is forced through the column by the force created by centrifuging, by the spinning, that liquid is collected in the collection tube. The plasmids, however, remain bound to the resin, so that's really good. Now here's a time to stop and look on the inside of your column. If your column still looks a little wet like mine does, you might want to centrifuge for just a little bit longer. So what I'm going to do is I'm just going to put my column back in, and I'm going to centrifuge this just a little bit longer. Now I'm going to adjust my grip on my centrifuge because my thumbs are getting tired. So I'm going to use my index finger now. As long as it's pointed away from you, it's fine. There we go, that looks better. I think mine just needed a little bit longer. So, yep, my column's nice and dry now. So, what we can do now, I'll just move that aside so you can see what I'm doing. I'm going to take my little waste beaker, and I'm just going to discard the liquid that flowed through our column. Okay, so we call that the flow through. Now we can put our column back in the collection tube, and we're ready for the next step. So for the next step, we're going to wash the DNA that's bound to the column resin. Okay, so remember the plasmids are stuck to that white resin in the column. So to do this, we're going to add wash buffer. So here's our wash buffer. I'm going to open up the tube. It's got a pink label. And I'm going to use a pipette. And I'm going to suck up all of that liquid out of that tube. And I'm going to put that liquid into our spin column. I'm going to discard this stuff. And I'm going to take my collection tube, or my column with its collection tube. I'm going to put it back in the drill centrifuge. And I'm going to run this for 2 minutes. Okay, so set my timer, and let's start. All right, so 2 minutes are over, so let's check the status of my column. Oh boy, so some of the liquid has gone through the collection tube, but there's still quite a bit left inside. So what that means is I need to centrifuge some more. And so you guys are just going to have to do this until all of the liquid flows through the column. Now, this might take me a few minutes, so I'm going to stop the recording, but what I suggest you do is do another 2 minutes. And then after that 2 minutes, check your column to see if there's any liquid in the upper part. When there's no liquid remaining in the upper part, then you're done. Okay? Okay, so I'm done. So that took me about an extra 4 minutes, and I realized that my battery on my drill was dying, so I had to go get my backup drill. So all of the liquid has gone through the column, and so I'm going to discard the liquid into my waste beaker, and I'm going to put my column back into the collection tube, and I'm going to put those back in my centrifuge. And now I'm just going to spin them for 1 minute, and this is going to help dry out the column in order to get rid of any remaining liquid that might be on that resin. So we're going to do 1 minute. I'll reset my clock, and there's our timer. Okay, so you may or may not see any additional liquid at the bottom of your collection tube. If you do, great. If you don't worry about it. Okay, and so now we have a nice dry column resin, and that resin has been washed, and so now we're on to our final step. In the final step, we are going to have to elute the DNA off of the column resin and into a microcentrifuge tube. Okay, so we're going to grab one of our micro tubes from our general supplies bag. Doesn't matter which one you grab. Okay. And what I like to do is take a pair of scissors, and I'm going to cut the lid off. And why I do this will make sense in a second. Now that I've just got the tube, I'm not going to throw the lid away. I want to keep that for the next step. I'm going to take my column out of the collection tube. Okay, that collection tube is dirty. And now in this step, we want anything that flows through our column to land in a nice clean tube. And so we're going to use this nice clean micro tube as a collection tube. So I'm going to actually put my column into that tube so we have a nice clean collection tube. Okay. Our old collection tube we can empty out into our liquid waste container, and then we can put in our waste bag. Okay. Now, to elute the DNA off of our column resin, we need the elution buffer. So this is the buffer, the final buffer in the little baggie, and it has a red label. Now all of my elution buffer is up at the top of my tube, just because it was laying upside down. So what I'm going to do is I'm going to flick it with my wrist in order to collect that liquid down at the bottom. And if that doesn't work for you, get your centrifuge out, put your elution buffer in with a balance tube on the other side, and just give it a quick spin in your centrifuge. And that'll force that liquid out of the lid and into the bottom of the tube. So now I'm going to take my last clean pipette out of the kit. And I'm going to use that pipette to suck up all of the elution buffer out of that elution buffer tube. And then I can take that and I can throw that away. So I'll just set that aside. I'm going to take that buffer and I'm going to add it to my column, okay, in its new collection tube. I'm just going to try to drop that liquid right in the center of that column, okay, just like that. I usually like to let this sit for 2 minutes in order for that liquid to really absorb into that column resin and to help loosen up the plasmid from being bound. So what I'm going to do is I'm going to set my timer to 2 minutes, and we'll just start that countdown. And so while that's going, I'm going to do a little tidying up. My empty tube of elution buffer I'm going to put in my bag. I'm going to use as my garbage. This baggie here, I don't need it anymore, so you can either save it for a garbage container for a later experiment, or you can put it in your waste container. It's had laboratory chemicals in it, so I wouldn't advise recycling it and using it for food. Your baggie of microtubes, well, we don't need those anymore, so we will put them back in our general lab supplies baggie. We're going to need our Sharpie. Actually, while we're waiting, we can label our tube with our Sharpie. And actually, I'm just going to write on the lid of the tube. Remember, I cut the lid off of the tube that we're using as our collection tube now. And so I think what I'll do is I'll just clean this hinge up, because that's going to bother me with that thing floating around. And I'm going to take that lid, and right on the top of that lid, I'm going to write P-glow, which is the name of the plasmid that we are extracting out of those bacteria. Hopefully you can read that okay. So now that I've done that, I don't need my Sharpie anymore, so I can put my Sharpie in my general lab supplies bag. And it's just about working neat and clean. It's kind of like if you're baking. It's always easier to bake when your space is clean and neat. Okay. Put my scissors aside. 7 seconds left. I also am going to need my drill for the next step. Alright, and there's our timer. So now I'm going to take my collection tube. I'm going to put that in my drill and put a balance tube right next to it. And we are going to centrifuge this for 2 minutes. Okay, and so let's do it. 2 minutes. Very good. All right, that looks great. So we're on to our final step. And in our final step, what we're going to do is we're going to take our column out of the tube, and we're going to use that pipette to suck up the liquid from the bottom of the collection tube. We're going to put our column back in, and we're going to put that liquid back onto the resin. And now we're going to centrifuge again for 1 minute. So I'm going to put that tube back in my centrifuge. I'm going to reset my timer for 1 minute. And this step just seems to help get a little bit more plasmid DNA. So I'm going to set my timer, and 1 minute. All right, very good. So, now that you've got the liquid down in the bottom of your tube, we no longer need the collection tube. So we can put that in the garbage. We can put the lid that we labeled back on this tube. And we can take this tube with our isolated plasmid, and we can store this in the refrigerator right next to the tube of pea-glow plasmid that we gave you in the original kit. Okay, so don't throw this away. As for everything else in the kit, okay, for activity 9, you can discard all of this in the trash. Your liquid waste container, you can discard down the sink or in the toilet and flush it away, and then you can wash this little cup with soap and water, and we can put it back in our general laboratory supplies. Okay, same thing with our pipettes. We can discard those in the trash. We can put our microtube blocks in our general laboratory supplies container and just generally clean up. And so I hope you enjoyed doing that activity, and we'll see you soon.